Hello everyone. Today I'm going to present my poster entitled Integrated Regulatory Network based on long non-coding RNA, miRNA, mRNA and transcription factor reveals key genes and subnetworks associated with dilated cardiomyopathy. This work was done by myself, Sona Charles, and my PhD mentor, Dr. Jayakumar Natarajan, in the Data Mining and Text Mining Lab, Bharatiya University, Coimbatore, India. Dilated cardiomyopathy is a multifactorial condition often leading to heart failure in many clinical cases. Due to a high number of dilated cardiomyopathy incidents reported as familial, a gene-level network-based study was conducted utilizing high-throughput next-generation sequencing data. We exploited the exome and transcriptome sequencing data in NCBI SRA database to construct a high-confidence regulatory network consisting of long non-coding RNAs, miRNAs, mRNAs, and transcription factors. We collected 12 samples of dilated cardiomyopathy and 4 samples of normal hearts to study differential expression. The reads were processed through the classical RNA-seq pipeline using HiSat, string-tie, and DE-seq. The batch effects were checked and normalized using Combat. The first figure shows the volcano plot of differentially expressed transcripts with a log fold change of greater than 2.5. The second figure shows the dispersion estimates, and the third one shows the MA plot of the transcripts. We identified the long non-coding RNAs by applying the conditions that the transcript should have at least 2 exons, the length should be greater than 200 nucleotides, and the FPKM value should be greater than or equal to 0.5. The transcript should not have coding capacity and should not coincide with other mRNAs or long non-coding RNAs. The bar chart shows the top 5 upregulated and downregulated coding transcripts and non-coding transcripts. We checked the chromosomal dominance of transcripts and identified that the number of coding as well as the non-coding transcripts were maximum in chromosome 1, that is depicted in the last figure. Combining the common targets of 20 miRNA target prediction tools, we identified 120 microRNAs that targeted 477 of the differentially expressed genes we have obtained through DE-seq. The gene ontology of the miRNAs and the differentially expressed genes are displayed in the figure. The geoterms of the genes were related to developmental process and that of miRNAs were related to gene expression and transcription. So this shows that there is a particular developmental process that is taking place or the rewiring of the heart tissues during the progression of dilated cardiomyopathy. And similarly, a gene expression change was taking place within the heart tissue that is contributed by the presence of microRNAs. Overrepresentation analysis was performed for differentially expressed genes in the pathways as well as for diseases. The overrepresentation analysis for diseases were done to identify any other possible disease associations with dilated cardiomyopathy. The age-rate signaling pathway in the diabetic complications was found to be the pathway with the highest enrichment ratio with 99 genes in the set. Hyperventilation syndrome and myocardial infarction were the 2 significant overrepresented diseases against the background list of OMIM. The long non-coding RNA interactions with miRNAs and genes were identified using star-based database. The network was constructed for interactions between long non-coding RNAs, miRNAs, and genes. The transcription factors in the network were also identified. The topological properties of the network reveal a scale-free nature. CIST, NOVA1, and MIR195-5P were found to be the hub long non-coding RNA, gene, and miRNA respectively. The MCL clustering of the network resulted in 5 modules with a score value of greater than 2. This module depicted here was found to be the only one having the presence of at least one long non-coding RNA, miRNA, gene, and transcription factor. Now, as a conclusion, the study helps in identifying promising targets contributing to the development and progress of dilated cardiomyopathy. The identified module consists of several microRNAs, long non-coding RNAs, and transcription factors, which is showing an involvement in cardiovascular diseases. So the identified module needs to be experimentally validated and has high potential to be a connected component of a pathway due to the presence of more than 5 nodes being involved in a cardiovascular disease. So thank you all for listening to my presentation. Feel free to ask any queries. Thank you.